Now, let us talk about aerobic respiration. Aerobic respiration takes place in the presence of oxygen. It occurs in three main stages: glycolysis, Krebs cycle, and oxidative phosphorylation. Glycolysis is the first stage of aerobic respiration. It occurs in the cytoplasm and does not require oxygen. It involves two main steps catalyzed by enzymes in the cytoplasm. In glycolysis, glucose is first activated by phosphorylation, meaning addition of phosphate groups. The phosphate is provided by ATP. During the process, two molecules of ATP are used and a glucose molecule is broken down into two molecules of triose phosphate. In other words, before energy is released by respiration, energy is needed to initiate the reactions. Next, each triose phosphate molecule is oxidized to a 3C compound called pyruvate. Two molecules of ATP are formed, and triose phosphate loses hydrogen, meaning it becomes oxidized. The hydrogen is accepted by a carrier molecule called NAD, nicotinamide adenine dinucleotide. As a result, NAD is reduced to form NADH. NADH will act as a hydrogen donor in the final stage of respiration to form more ATP. The breakdown of a glucose molecule into two molecules of triose phosphate consumes two molecules of ATP. The oxidation of the two molecules of triose phosphate forms four molecules of ATP. Therefore, glycolysis produces a net amount of two molecules of ATP. In summary, during glycolysis, a glucose molecule undergoes oxidative breakdown into two molecules of pyruvate with the production of two molecules of NADH and two molecules of ATP. Therefore, the overall equation for glycolysis is glucose going into pyruvate using 2 NAD to change to 2 NADH, and 2 ADP is phosphorylated to 2 ATP. With pyruvate, a large proportion of the energy of glucose is still trapped in it. Therefore, pyruvate will be further oxidized to release more energy in subsequent stages. A reaction bridges between glycolysis and Krebs cycle. The pyruvate formed in glycolysis then enters the mitochondrion, where the Krebs cycle occurs. Pyruvate does not enter the cycle directly. It is first converted to a compound called acetyl coenzyme A, or simply acetyl-CoA. During the conversion, a carbon atom is removed from pyruvate and released as carbon dioxide. The remaining 2C compound loses hydrogen, which is accepted by NAD to form NADH. It then combines with coenzyme A as a carrier molecule to form acetyl-CoA. Enzymes in the mitochondrial matrix catalyzes the conversion. For each molecule of glucose that undergoes glycolysis, two molecules of acetyl-CoA are formed. Acetyl-CoA will donate its 2C acetyl group to form a 4C compound in the first step of the Krebs cycle. The Krebs cycle occurs in the mitochondrial matrix. It involves two main steps catalyzed by specific enzymes in the matrix. First, the combination of acetyl-CoA with a 4C compound. Secondly, the regeneration of the 4C compound. Step 1 is the combination of acetyl-CoA with a 4C compound. Acetyl-CoA, a 2C compound, combines with a 4C compound already present in the mitochondrial matrix to form a 6C compound. In this way, coenzyme A is regenerated continually for carrying other acetyl groups formed from the pyruvate into the Krebs cycle. Step 2 involves the regeneration of 4C compound. The 6C compound is then oxidized step by step to regenerate the original 4C compound. During the process, 2 carbon atoms are removed and released as carbon dioxide. The 6C compound also loses hydrogen. The hydrogen is accepted by NAD to form NADH. Another carrier molecule called FAD, flavin adenine dinucleotide, also accepts hydrogen to form FADH. In addition, 1 molecule of ATP is formed. Therefore, in summary, in each cycle, 3 molecules of NADH, 1 molecule of FADH, and 1 molecule of ATP are formed from 1 pyruvate molecule. Since each glucose molecule generates 2 pyruvate molecules, a total of 6 NADH, 2 FADH, and 2 ATP are formed from 1 glucose molecule. The ATP, NADH, and FADH formed store the chemical energy of glucose. The energy stored in ATP is readily usable by the cell. The energy stored in NADH and FADH will be released in the final stage of aerobic respiration. The final stage of aerobic respiration occurs on the inner membrane of the mitochondrion. It is called oxidative phosphorylation because the energy for forming ATP by phosphorylation comes from oxidation of NADH and FADH. Step 1 is the regeneration of NAD and FAD. NADH and FADH formed in glycolysis and the Krebs cycle lose their hydrogen and are oxidized to NAD and FAD respectively. By losing hydrogen, NAD and FAD are regenerated. They can accept hydrogen in glycolysis and the Krebs cycle again. Step 2 is the splitting of hydrogen atoms. The hydrogen atoms split into hydrogen ions and electrons. Step 3 is releasing ATP through a series of redox reactions. The electrons take part in a series of redox reactions catalyzed by enzymes embedded in the inner membrane of mitochondria. Energy is released to form ATP in the reactions. The hydrogen ions and electrons are eventually transferred to oxygen, the final electron acceptor, to form water. Therefore, in summary, during oxidative phosphorylation, oxidation of each NADH forms 3 ATP, while oxidation of each FADH forms 2 ATP. As a total of 10 NADH and 2 FADH are produced from glycolysis to the Krebs cycle. The total number of ATP formed in oxidative phosphorylation is 10 times 3 ATP from NADH plus 2 times 2 ATP from FADH equals to 34 ATPs. Now, let's talk about the overall summary of aerobic respiration. The energy yield in aerobic respiration. In aerobic respiration, part of the energy released is stored in ATP, NADH, and FADH. The energy stored in NADH and FADH is finally transferred to ATP during oxidative phosphorylation. The total number of ATP formed per glucose molecule is summarized here. In glycolysis, 2 ATP and 2 NADH are formed. Therefore, the total ATP formed in glycolysis will be 2 directly from ATP and 6 ATP from 2 NADHs. Therefore, glycolysis in total gives 8 ATPs. During the conversion of pyruvate to acetyl-CoA, 2 NADHs are formed. Therefore, in the end, 6 ATPs will be formed. During the Krebs cycle, 2 ATP, 6 NADHs, and 2 FADHs are formed. Therefore, 2 ATPs are formed directly. 18 ATPs are formed through NADHs, and 4 ATPs are formed through FADHs. In the Krebs cycle, a total of 24 ATPs will be formed. Therefore, the total number of ATP formed in the complete breakdown of one glucose molecule is 8 plus 6 plus 24, which is 38 ATPs. The overall equation of aerobic respiration: 6 oxygen molecules are required to break down a glucose molecule completely. 6 carbon dioxide and 6 water molecules are released in the process, and 38 ATPs are yielded. Therefore, the overall equation of aerobic respiration is 1 glucose molecule plus 6 oxygen molecules goes into 6 carbon dioxide with 6 water molecules and 38 ATPs as energy. Now, let's talk about why oxygen is so important in aerobic respiration. Oxygen is only used in oxidative phosphorylation. It acts as a final electron acceptor. If there is no oxygen, electrons released from NADH and FADH cannot take part in the series of redox reactions. Oxidative phosphorylation will stop. NAD and FAD cannot be regenerated. As a result, the Krebs cycle can no longer operate, and only glycolysis can continue. The number of ATP formed will be greatly reduced. This is the end of the video. Thank you for watching. Please give us a like and subscribe to our channel. Click on the tab to watch the next part of the video series.